Bismillahirrahmanirrahim. I welcome you all for today's lecture that is on methods in neuroscience. And in this lecture, I will try to describe the methods which are commonly in use for different brain studies and what are their possible implications. The research in neurosciences is progressing very rapidly, and there are a lot of new techniques and advancements, and they have a lot of implication, as you will see during the lectures as well. Before this, I will start with one example that, for example, if you have to take out the money from ATM machine, the techniques are so like powerful. This is just an example. And initially, we used to put the card inside, and that is one way of getting the money. And recently, the thumb impression is being introduced. That you just put the thumb impression, and then you can take out the money as much required. But if even we use a thumb impression, that can be like copied, somebody can copy that. So there are chances that somebody misuse your ATM. Another powerful technique is the retina scan. So another approach is that is also being used at some of the security places is the face scan, and especially the retina scan, the scan of your eyes, that is specific for you. And that can also be, there are chances that that can also be misused. But in neurosciences, the techniques are so powerful that in future there could be a brain scanner is there, a machine in ATM machine, and there if you go inside and they ask for a password, and you just remember your password in your brain, and simply it will work for you. So the information from your brain, they will be decoded, and those information because the specific waves are released from the brain, so the information will be decoded, and your ATM will be, and they can even check your stress level or anxiety level. So if you will be, for example, if there are some person outside and he wanted to uh, get the money from you, and you are so stressed, or somebody is you are inside and somebody is trying to move inside with the bad intention. So in that case, if you are really stressed, then that machine can also guess your stress level and can also ring a call to the police station. So these techniques are so powerful, and similarly, they can be applied for education. For example, I will give some of the example during the lecture as well. That during, if you have to open your, for example, a laptop or some other machines that you want to use. So in future, it could be your just brain thinking, that you just think about something and the things will be open for you. So these are so powerful techniques. So in this lecture, I will go through some of the techniques which are being used. We will discuss the techniques starting from the neuron, how we can study the neuron at the cellular level. Then we'll discuss the slices that we make of the brain. For example, we take the brain out from the model animals and we slice them into very thin pieces, and then we study. That is another approach. And then we'll also discuss the brain scans, that will do different type of brain scans, and what are different approaches to do the whole brain scans, and what implications they have. And then we will also discuss the model animals studies, how we do the studies in model animals, and we do the genetic manipulations and so on. So it will be very interesting for all of you to learn some basic technique in neurosciences that we use and the implication they can have. So I will not discuss in detail all these techniques because each technique need a whole lecture to describe. So it will be just an overview, so you can have an idea that these are the techniques which are being used at different levels to investigate the brain. And my details are given. So if you have any questions, you can definitely email me, or you can WhatsApp me, because my WhatsApp number is also given uh, in the group that we have, and the group about the educational neurosciences. So these techniques, as I was mentioning, these include the cell studies, cell work, to study the study brain cells or slices, and then we'll discuss measuring brain activity in living organization. organisms that are the scans for functioning brains, how we can check the activity in specific parts of the brain. Like if somebody is happy, we can see the changes in the player center of the brain. If somebody is sleepy, then we can see the inactivity in the sleep center or the activity in other portions of the brain. And then implanting measurement tools and measures. So if we have to implant something inside the brain, I will also discuss some of these approaches in which we do the implants in the brain to check the brain activity, the electrophysiology of the brain, of the neural networks. So we'll discuss that, and we'll discuss the animal models and also post-mortem examination. So these are the post-mortem examination of different animals as well as the human. So in case of human, for different neurological disease like Alzheimer's, Parkinson's, and so on. So we have to study the brain. We take out the brain after the death of the person, and then we investigate that what changes, what physiological changes are there in the brain. So these are different approaches that we use in neurosciences. So we are going to start with the staining and imaging the neurons initially. So I will describe these briefly. I will mention the technique. So one is the Golgi stain method that is being used for the studies of the neuron. And it randomly stains about 5% of the neuron in slice and place them in relief against the background. And you can clearly see these neurons. And also you can see the specific pattern, because in brain, as I mentioned in the previous lectures, that if some activities is going on and you learn some skill, then there is a specific neural network that you can study, that these are some specific pathways in the brain that are there, and these are the very strong pathways as compared to the other pathways. So this is one of the method. And for slice, I would like to mention that brain is a very delicate organ. So if we have to study the brain slice, then the approach we mostly use is the cryo slicing approach. So we prepare just like the microtome we use for the slicing purpose. But in this case, because the brain is very delicate organ, I used to do the slicing in Japan. So in that case, we used to put the slices in a chamber that is super cold. It was freezing inside, so we use specific gloves, move inside, and then we do the slicing. So these are very different as compared to other organs like the liver or the heart, because the brain is delicate, so we have to prepare the slices very carefully. And so the Golgi stain method is there, and then there is myelin stains. So myelin, are the structures that surround the axons of the neurons. So the conductivity of the neurons can be enhanced, and it shouldn't be interfered. These are rich in the fatty acids. So stain taken up by the fatty myelin that insulate axon, and stain help identify neural pathways. So once again, as I mentioned in my previous staining method, that there are specific neuronal pathways that we can study in this case. So these are the neurons, they wire together for some specific behavior or for some specific reaction. And then I used to do commonly this nasal stain. This nasal stain actually is to stain the cell body. So if you have a brain slice, for example, and you want to see the cell bodies, this nasal stain can also be used for other organs like for the liver or for the heart. And this can also be used for the brain. So we do the nasal staining. There is a specific staining method that you can check online. Nasal stain is also one of the approach that actually identify the cell bodies of the neuron. So there is if you study the neuron, then there is a cell body, and then dendrite and axon and other structures. So this nasal stain is specifically for the cell body. And then there are also approaches for autoradiography. So there are different approach in which we use the radiographic material. For example, we use the fluorogold. This fluorogold make neurons stand out importantly, tell which neuron are active, and can correlate with behavior. So this is very important, because in this case, we can see that which neurons are active. So this fluoro gold, they also use for the staining purposes, and then we can study the neuronal pathways and activity of the neuron. In addition, this is also very commonly used, the 2-DG, 2-deoxyglucose. This is the radioactive form of the glucose because, as I mentioned in the previous lecture, that brain is very active in energy because it's just 2% of the body weight, but it consumes 20 to 25% of energy. So it takes a lot of energy. And if we label the glucose, which is readily taken up by the neurons, then we can see specific regions in the brain where there is more consumption of glucose. Wherever there will be more activity, there will be more uptake of glucose, and hence we can study that these brain areas are more active. So this is one of the approach, the 2-deoxyglucose, and make this sugar radioactive, and it taken up by the neuron can trace where it went. So as I mentioned that we can trace where it has went and where the brain activity is going on. In addition, they can also stain for neurotransmitters and other brain chemicals. You know, there are so many neurotransmitters which are involved in transmission of the signals between the neurons. So we can also stain those neurotransmitters and can specifically check that which neurotransmitter is active at this point. And then we can also see the behavior, the linkages of those neurotransmitters with the behavior. So this is also, so there are different staining that can stain different parts of the neurons. And then we can also use the auto radiography approaches for the brain. In addition, we can also use light and electron microscopy. We can use the simple microscope and we can stain these slices and the neurons. We can grow them in the culture as well, and then we can stain them. But we can also use electron microscopy if we have to study all the neurons in very specific details and what changes has happened in case of any disease or in any pathophysiological condition. Then we can study through the electron microscope. So in electron microscope, what happens that passes beams of electron through thin slice of brain tissue onto photoplate, and different parts of tissue block or pass electron at different degree, and electron produce image based on this variation. So images are produced, electron microscopes are being used. I used to do this in Japan, but there are now very so much different approaches are being used. For example, now there are cryo-electron microscopy that is also being introduced in different countries of the world, and these are very powerful microscope because otherwise we have to prepare the samples and it takes a lot of time to investigate just a single sample. But in cryo-electron microscope, you can also operate them from home. You can put just the sample and it is taken up in a specific way, and then you can put multiple samples and you can investigate even from the home. So these are the powerful microscopes. Then there is another microscope, is the scanning electron microscope. So scanning electron microscope, basically, in this case, the light doesn't pass through, but it scans the surfaces of the cell. So a beam of electron cause specimen to emit electrons itself, and these are captured by the photoplate, and then they are superimposed on each other and produce the 3D image. And in this case, we can see the images just like this of these electrons, that how they are connected to each other and what dendrites are there and what cell bodies are there and what is the sizes of these neurons and so on. So these are very powerful approaches that are being used for the studies of the neurons. And then brain activity, we can measure the brain activity through electroencephalography or EEG. This is a very old technique introduced very long ago, and they can actually they can decode our brain activity, and this is very powerful technique, and they have a lot of implications. And in the future lecture, I have some very interesting slides in which they have measured the EEG in real time during the lectures, and they have seen that what is the difference during the lecture or the lab work or the homework, during watching TV or during the sleep. And these are very interesting images, and I will definitely show those images, and they have a lot of implication in our educational neurosciences. So these are EEG. They can record from the 2 electrodes on skull over area of interest. So wherever we want to see the activity, we can put the electrodes, and we can measure the brain activity through the brain waves they are released. So they uh, brain waves, they are taken up by the electrodes, and then they produce some specific graphs. These electron amplifier detect combined electrical activity of all neurons between these two neurons and can graph the activity. So in this case, we can produce the graphs just like this, whether there is more brain activity, whether there is a straight line, whether there is a less activity. So these are read by some specific experts of the field, and they can give the implications like whether the brain is active, whether brain is not active, they are taking interest or not, and why they are used. And they detect changes in brain pattern or arousal and can average several readings to obtain evoked potential. And there could be some background noise, but now the techniques are so powerful that we can omit the background noise, and then we can have a better estimation of the patterns. So in this case, we can actually have the brain activity, and they have a lot of implications. As I mentioned in the previous lecture, that even in China now, based on the 5G technology and through the artificial intelligence and through scanning the brain activity, they have put some smart gadgets on the brain. You can see over here, these are gadgets up there. And these gadgets, they can measure the brain activity. So these gadgets, they release the data, and that is coming on the monitor of the teachers, and they can observe in real time whether the students, they are taking interest or not. So if you are giving a very boring lecture, then there will be less brain activity and less concentration, and you can have a guess, no, the students are not taking, there is need of some break, or I should tell them some story, and so on. So they have a lot of implications, and the future like uh, classroom will be look like this. So you can imagine that you have some variable in the university students and in the college students and the school students, and you can see whether in real time whether they are taking interest in your class or not. So these are very powerful techniques. Then there are stereotaxic techniques. So I will describe briefly these stereotaxic techniques. They have, uh, we use some stereotaxic devices for this, and this allows precise positioning in brain of electrode and other devices. So in this case, we just, for the animal, we have to unconscious it, and then we open the brain, and then we put specific, I used to do while I was in Japan. So we used to, I have one slide on the technique that we used to do, as I also mentioned in the first lecture, that the area that we were working. So in that case, we used to put the devices, and we used to see the brain activity in the brain. So in this case, we introduce electrodes inside the brain, and then we hold head in a position. So we just hold it in a position, and we can 3D height, depth, and width. So we have the idea that we take the brain atlas, and then we compare it, and where we have gone, and what is the region, what it is, whether it is hippocampus or whether it's thalamus, or what area of the brain is there, and then we check the brain activity. So in this case, we can, several kind of things might be put into the brain. These could be a cannula, these could be electrode, and these just like this. So they can be introduced in specific regions of the brain. They could be like related to the memory, touch, taste. So whatever we want to investigate, we can introduce there, and then we measure the brain activity in those regions of the brain. So in this case, it allows one to ablate or lay in precise area of the brain. So in this case, we can also, like we can, through the electrodes, we can ablate some specific neuronal activity. So just like in case of epilepsy, there is some areas of the brain, there is activity, and we have to ablate that activity, so we can neutralize those activities in the brain by introducing some spice. So these techniques is also being used in humans now. In this case, they introduce the deep brain electrodes, and they can alter specific brain activity in the patient. So in this case, they alter specific area to determine function. So the one way is to, if we want to know some specific neuronal network, what implication it has, and what role it is playing in the brain. So we can actually activate it or we can stop it, and we see the behavior of the animal and see what is the role of this. And there is now the human brain project is going on in United States and also in the European Union, in which they want to give some specific, there are around now 100 billion neurons, so they want to give some specific function to each neuron in the human brain project. So it's a huge project. So we can also, like, these are the approaches in which we can assign different functions to the brain. So there are several kind of measures. We can do the electrophysiology, we can do electrodialysis, and we can also do fast scan, cyclic voltammetry. So we can do all these kind of things. And in this case, we can check whether there are some electrical or chemical changes in the brain. They are there. So we can, as I mentioned, that we can use the electrodes or we can use cannula. We can take the sample, we can measure the chemical changes in the brain, we can measure the electrical changes in the brain, we can ablate the function, we can introduce the function, we can activate some neural network, we can block some neural networks, and hence we can check the brain activity. So these are different approaches are being used. So that those approaches were the stereotaxic, and now we are moving towards the brain imaging. There are different brain imaging techniques. Initially, they were not that powerful, and we used to get very raw sort of images of the brain, but now we can get very powerful images of the brain, and also we can monitor the brain activity. So I will quickly go through these techniques. One is the initially, I think in Pakistan, like around 10 years ago, there were the CT scan, and before this, people only go for the X-rays. But then CT scan was, or the CAT scan was introduced. This is computed tomography, and this is based on X-ray scanning. It produces series of X-rays taken from different angles. So in this case, this is more powerful because you are inside the machine and it takes the X-rays goes in different angles, and then the computer help in creating 2D horizontal cross section of the slice, and then these cross section, they are computed together to make the 3D images, so we can make the 3D. Before that, in case of X-ray, we just used to get the 2D images, but in this case, we can take them several angles of the brain and in the machine, and then we can make through the computer the 3D images and can have a better idea of the brain structure we want to investigate. And then another technique was introduced, that is MRI. MRI is more powerful. After the CT scan, MRI was introduced, I think around like around 7-8 years ago, maybe more than that. So this is MRI is the magnetic resonance imaging, or MRI. It's measure the radio frequency waves emitted by hydrogen atom when they are subjected to strong magnetic field. So a strong magnetic field is applied, and then hydrogen atoms frequencies being monitored. This is very fast, and it can scan very small area very clearly. So you can see this MRI scan, this is some specific regions of the brain. And if we want to investigate what changes has happened in these specific regions of the brain, just like this, we can take the image from MRI and we can investigate, okay, these changes has happened. So this is a very powerful technique that is being used in neurosciences, and it has a lot of implications. Then there is a PET scan. So MRI, and then there is a PET scan. PET is positron emission tomography. So that is called PET. So because there is emission, so it's based on the radioactive material. So it involves injecting radioactive substance into the bloodstream. It is taken up by the parts of brain according to how active each area is. So as I mentioned that some specific, if there is some specific function, for example, you are listening a lecture and your memory areas are active now, so we can check through PET scan which area of your brain are active. Because you are watching this, your occipital lobe will be active and your memory portion will be active and so on. So these are the techniques which are being used for the investigation. So PET scan is often we use the radioactive in as I mentioned, that 2-deoxyglucose is used, and we can use other radioactive traces to mark blood flow or the oxygen uptake. So we can use multiple radioactive material in case of PET scan. It requires a lot of training and access of cyclotron. So you put the person in a cyclotron, and then you need the training, and then you have to supply the radioactive material, and it provides estimates of brain activity and changes in brain activity. So it gives you just like this. Some areas are very active in case of brain, and some are not that active. So the PET scan, uh, either oxygen uptake, because brain also takes a lot of oxygen, brain takes a lot of glucose, so we can check which area of the brain are more active using the PET scan. And this is like if you are going to study some people, and during some specific, for example, you are the person is inside the cyclotron, and they are watching they are trying to learn some specific technique. For example, they want to learn how to play the guitar, or they want to they are inside and they are playing football, for example, football game, for example. So in this case, we can see that what actually brain networks are active in that case, and what regions of the brain are active in that case. So these are very powerful techniques which are used for the investigation of the brain. And then we have investigated one was the MRI, magnetic resonance imaging. And this is another very powerful technique that is being used nowadays is the functional MRI. And this is a lot of use in educational neurosciences is the functional MRI. fMRI, functional magnetic resonance imaging, this is another technique which is used. So if we have to study some specific function of the brain, we use this technique. It measures brain activation by detecting increase in oxygen level in active neural structures, and can be used as individual is engaging in behavior or cognitive tasks. So this is, as he mentioned, that these are the techniques which are in a lot of use. So especially in educational neurosciences. So if you are doing some numerical, which part of the brain is active, if you are watching some specific structures like the triangle or the circle and so on, which electronic, which brain networks are active in that case, and so on. So, or if you are doing some calculations, which part of the brain will be active. So these are very powerful and very fast techniques. And in this case, these are the non-invasive, because in this case, we are not going to introduce the radioactive material, and it gives it gives the details of the structure of the brain. And this is in large use in education neurosciences, can be used in the cognitive task, and can see changes as behavior change. So we can also, in real time, we can see a change in the pattern of the neural networks. And these are the techniques in which can decode the info. So after fMRI, we can in real time decode the information from the brain. Even like somebody is thinking about something, we can decode the information through fMRI, that the person is thinking about some event, or he is thinking about. So in future, might be if you are thinking, or if you are dreaming, then we can decode your thinking, we can decode your dreams and so on. So this is one of the approach, and even more advanced approaches are there to know the brain activity. So we can, in real time, we can monitor the brain activity and links some specific. Before this, like brain was thought to be that some areas are linked to this, but now we can pinpoint some specific behavior with some specific areas of the brain and the changes we observe in that area of the brain. In addition, one of the approaches is to study heritability and genetics. So the genetics, another approach in which we use the family studies, we use the adoption studies, twin studies, and concordance rate. So I will briefly describe some specific functions. Some genetic diseases could be there that are passing from family to family. Some features could be there like intelligence or some other features. So we do the family studies. And in family studies, we determine how strongly a characteristic is shared across family members. What is the quantity? and correlate degree of heritability. So how much it will be inherited to their children. So in this case, we check the characteristics strongly which are shared among the family members through the family stories. Then we also do the adoption studies, compare adopted and biological children. We can have a comparison of these and compare behavior in adoptive versus biological family. So this is also a very powerful approach in which we use the behavioral studies, and then we link it through these powerful machines. We further investigate those. And then there are also twin studies. Either they are identical are fraternal twins. So we also do the twin studies of, and we also see what behavior is common among the twins, what are not common among the twins, and so on. In addition, we also study the concordance rate, that is the frequency with which relatives are alike in characteristics. So how much you are alike with your relatives. If your relatives are very generous, for example, are you the same very generous person or not? Some like just like the people say that Maiman or the sheikhs, they are very good planner for the business. So either you are a good planner of the business or not. So these are the things that we actually study through the heritability and the genetic studies. And now this is the era of genetic engineering. So in genetic engineering, we can manipulate of organism genes or their functions. So similarly, we can also manipulate the genes, and then we study how much impact it has. Some genes are very important for the brain function. So if you lose those genes, we used to, when I was working in Japan, another lab was working on some genes which were very important for brain function. So when they knock out those genes, there was no brain development and there was early death. So some genes are very important. So we also study that what is the linkages of these genes with some specific characteristics, some specific behavior, and also what is the role in the developmental processes. So this is also very important area of learning in which we do the genetic engineering and do the genetic manipulation. Knockout techniques are, knockout is mainly that is used for the genes in which we use knock out or remove some specific gene, and then we see the behavior, the effect. This is mainly done on the model organism like Drosophila, which used to do in the mouse, and we ablate or knock out some specific genes, and then we see what happens. Either there is some developmental defects or there is a defect in the behavior. For example, uh, there was a very interesting study in which they ablate one of the gene, and then they showed a very different behavior, and they have a lot of attraction towards alcohol in flies. So these are very interesting studies in which they do the knockout techniques. And then there is antisense RNA procedures. In this case we are not actually knock out the genes, but we uh, stop the function of the protein. So in this case, the messenger RNA is produced, and then we use antisense to those messenger RNA, and we block those messenger RNA, and they are not translated, and hence no specific behavior or function is achieved in this case. So this is also very interesting technique. I used to do this technique back in 2008 when I was doing my research in United States. In that case, we used to use these antisense RNA in some specific gene function. We used to study the differentiation of cardiac stem cells into cardiomyocytes, and we use some of these antisense RNA to see whether we can block some specific genes and how we can alter the functions. So this is also approach, and then we can also do the gene transfer. For example, this glowing rabbit. In this case, we can introduce the green fluorescent protein into the rabbit. They are not there into those organisms. We can introduce those genes during the embryonic development when there is a blastocyst formation, and then we can introduce those genes in embryonic stem cells, and hence they are transferred to the whole organism, and then we can see. And in addition, we can also give some specific functions to the brain. We can introduce some genes which can only expressed in the brain, and we can see the effect on the brain. So this is also a very powerful technique. Then we can also go for the transgenic animal. Transgenic animals are those in which we introduce a gene into those animals. They will be the transgenic animals. And in this case, we can also see whether we are going to express those genes in specific regions. We can express those genes in the brain. We can put some specific promoter region that is only expressed in the brain. So we can introduce that. We can express those genes in the brain, or we can express those. For example, I was in Iran 2 years back, and they have the transgenic animals which were expressing growth factors. So they introduced trans genes into the animals, and they were expressing in the mammary glands, and hence the growth factors they were released along with the milk. So this is how we can also do the transgenic. We introduce the genes into the animal and express some specific genes in those. And genetic engineering, now there is the new technologies. Initially, it was a very time-taking process to do the genetic manipulations, and everything was like time consuming and expensive. But the new techniques like the CRISPR-Cas9 system and even the more powerful techniques are being introduced afterwards. So in this case, we can easily manipulate now the genes into, and we can manipulate the functions of, and then hence we can investigate the specific regions of the brain if we introduce some genes, or if we ablate the function of some genes into the brain, and then we can observe the specific behavior in those animals. And this is a technique I was working in Japan, and this is called the optogenetics. Opto mean light, and genetics mean genetics. So in this case, we use the light to uh, actually make the changes in the neuron. So this is a very interesting thing. Actually, in this case, we introduce the genes which are photoreceptor. For example, the channel rhodopsin protein, CHR2. So we introduce this channel rhodopsin, and then we can activate this channel rhodopsin with the specific wavelength. And if we can activate, then we can activate some specific neuronal circuit, and we can achieve some specific behavior. For example, just to simplify the things, if you are, this is a lunchtime or post lunch time, and you haven't had your lunch, and you are very hungry, and actually your hunger is controlled in specific area of your brain. And if we do this manipulation and we activate the brain area with specific light, in milliseconds we can finish your hunger. So this is how powerful. If you are feeling sleepy, sleep is controlled by the brain. In milliseconds, your sleep will be gone. Or I think the area we were working was the depression. So depression is actually because it's a very lengthy procedure in which there are changes in the brain, and our player center is inactivated. So we can activate the player center of the brain in milliseconds. So otherwise, we have to take the medicines for a very long time to be a normal person. But through this technique, in milliseconds, a person is depressed, and we can activate the player center. Similar to channel rhodopsin, another approach is it's another way is to actually silence the brain. So in this case, one way is to uh, activate the brain neuronal network, that is to generate the action potential. And another way is another set of protein if we introduce into the brain, then we can actually silence the action potential. So if there is action potential, we can we can stop the action potential, and hence we can stop the signal. So this is a two-way approach. We can activate, or we can inactivate some regions of the brain. So these are very powerful techniques that are being investigated, and it has a lot of implication, because this is a very powerful technique in which we can introduce the channel rhodopsin into the brain, and by putting some specific light, we can have some specific functions of the brain. So this is very powerful technique to know some specific regions and their linkages with the behavior, and some specific neuronal networks that are in our brain, and how they can be activated, and they can be linked to some specific behavior. So these are very interesting techniques. I hope you have a very good idea about a broader idea about different techniques that are being used. And these we will also like introduce during our lectures, that what techniques they have used to investigate this a particular finding, and what implication it has. So it is very important to learn these techniques initially, and that how we can use the single cells, how we can do the staining, how can we do the autoradiography, how we can do the brain scans, and how we can do the genetic manipulations, and we can have some specific function. So now we talk about, there is, because brain is very important organ. If the brain is changed, your behavior is changed, your whole personality is changed. So there is a lot of ethical regulation related to the brain research, and you have to go to whatever research you are going to do. You have to pass through the regulatory uh, organizations. That could be regulatory body of your institute or university, or that could be at the government level. So there is regulatory body for the animal research that we have to follow their guidelines. And then there is also problem during this research of plagiarism and fabrication of data. And they have very actually bad implications, because if you start if you introduce something in your classroom based on the data that is fabricated, that is a false data, that is a very wrong thing, then you will not get the desirable result. So after going through this course, you will have a better idea that whether this is a valid technique or not, because after going through these lectures, you will have a very good idea about that, what techniques are there, what is their implication. So if somebody is talking about something which is you consider that this is not possible, and it could be a false data, then you can easily discriminate now on the basis of the knowledge you will gain through these lectures. So, and then there is an ethical dilemma about the gene therapy and stem cell therapy. There is a lot of discussions are being there whether we should do the manipulation in the brains or not, especially if we talk about the human. If we introduce some changes in the human brain that can have a long-lasting implication. So there is a lot of discussions, and all the stakeholders should be involved to resolve these issues. With this, thank you very much for your careful listening of this lecture. I hope you will have very good idea about different techniques that we use. If you have to go through this technique and you have some problem, then definitely you can discuss with me, and I will try to elaborate. As he mentioned at the very start, that each technique, even we talk about CRISPR, for example, or if we talk about the gene knockout, and I give you some specific examples of gene knockout in the brain region, then it will take the whole lecture on each subject. That's why in one lecture, I have given a broader overview that what are different techniques which are being used for neurosciences, and what implication they have. So this is a broader perspective. If you have to study, you can also Google it, and you will get a lot of data about these techniques. Or if you have any problem, then definitely you can also ask me. One thing I mentioned in the orientation session, that we will also have some assignment. Hopefully, I will ask the coordinator to send you the assignment either today or tomorrow. So you have to complete these assignments as well. These are not very tough. This will be a just single slide assignment, and you have to prepare. So you will have a good like we should knew that you have gone through the lectures and you have a very good understanding about these techniques and about implications and so on. So we will also share with those. Once again, if you have any problems, you can discuss with the coordinator or myself, and we'll be happy to answer. Thank you. And then tomorrow we will be for the next lecture that is very interesting about the neuromyths, and hopefully you will find that interesting as well. Thank you.